Hello students, in my last video we have started with Unit 8 Biology in Human Welfare. In the Unit 8 chapter number 10, that is Microbes in Human Welfare, we have started in my last video and we have finished with microbes used in the household products. We have seen how different microorganisms are going to be used into our day to day life, right from the making of the cheese to the curd to the fermentation of the idli and dosa batters, or even in many other household products, we are using the microorganisms. So, that was the microbes in the household products. Today, let us see how these microbes are going to be produced at an industrial scale, or microbes used in industrial products. So, when I am talking about industry, remember industry means nothing but large scale production, producing on in a more extent or producing in a larger quantity is going to be the industrial production. So, how these microbes are playing an important role in industrial products, let us see that with some important examples. Coming to microbes in industrial products, we can see that the production of the beverages, antibiotics, etc., on an industrial scale requires growing microbes in very large vessels. So, when we are talking about use of the microorganisms on an industrial scale, means on a large scale. So, in a large scale, we cannot grow the microbes in a petri plate or in a test tube. We have to grow them without contamination, maintaining proper conditions in a larger scale. So, at that time, we are using something known as the fermenters, as you can see over here. These are going to be called as the fermenters, in which the fermentation by the microorganisms is going to be done. What is fermentation? Fermentation is nothing but producing the products from the microorganisms. For example, simple, if you are going to be producing your curd, curd is also going to be a product of fermentation. But producing of the curd in large extent, suppose if you are going on the packaged Aditya milk or any Nandini, you get the packaged curd. So therein they are not putting the starter culture, or they are not putting the curd into normal vessels. They are using these type of fermenters to grow the microorganisms and produce the product in the controlled condition. So those are known as the fermenters. So in these fermenters, the microorganisms are going to be grown on a large scale. Then here, let us take some examples. Fermented beverages are mainly produced by the use of microorganisms in industries. Here you can see that the Saccharomyces cerevisiae, also known as the baker's yeast, and now here it is known as the brewer's yeast because it is used in the production of beverages by fermenting malted cereals and fruit juices to produce ethanol. So these are going to be the yeast, that is your Saccharomyces cerevisiae or the brewer's yeast, what they call, and these are going to be your malted cereals. Malted cereals are nothing but the cereals are, first of all, your dals are known as the cereals. So small seeds whichever are going to be there, leguminous seeds are known as the cereals. So here the cereals with the covering are let to grow what you can say, they are led to sprout out a little. You can see here the white color portion is going to be coming out. So these malted cereals are fermented, or even the fruit juices are going to be fermented using the yeast, that is Saccharomyces cerevisiae, to produce ethanol. And ethanol only is nothing but your alcohol. So in this manner, in the production of the wine and beer, we are using them, that is the wine and beer are produced without distillation. So they are crude beverages, you can say. And whereas the whiskey, brandy, rum or gin or arrack, etc., these are going to be produced by the distillation of the fermented broth. They are allowing the fermented broth, what is there, they are allowing to boil it, and the wafers that are coming out, they are condensing it, they are cooling it, and those wafers they are collecting, which is the concentrated ethanol or the alcohol, and that they are going to be transforming into various things like the whiskey or brandy or such things. So that is how the fermented beverages like your wines and beers are going to be produced in the industries using the microorganisms. Then apart from that, we can see that the microbes in the industries are mainly used to even produce the antibiotics. Now first of all, what do you mean by antibiotics? Here anti means against and biotic means life. So antibiotics means against life. So antibiotics are chemical substances produced by some microbes and can kill or retard the growth of pathogens. So the antibiotics are already produced by using some microorganisms only. But these chemicals that are going to be produced, these are helping to grow or helping to kill other microorganisms or other pathogens. So they are against other pathogens' life. So hence these antibiotics are going to be a very important part of the human health, as they are used to treat many diseases, many dreadful diseases like plague, whooping cough, diphtheria, leprosy also, like diseases are going to be cured by using the antibiotics. Coming to the discovery of the first antibiotic discovered was the penicillin, and it was discovered by Alexander Fleming. Now there is a story behind this, a very you can say story which you should know. So what happened, this Alexander Fleming was a Scottish researcher. He was working in a hospital as a lab technician, as a researcher, and there what happened, he was known as a very careless lab technician. So he used to not take proper care of the laboratory as such. So what happened, one day he was experimenting in the petri plates. In the petri plates he was growing the streptococci bacteria, that is the cephalococci bacteria, and he just left those plates like that only, unwashed plates he kept like that only, and he went for leave for around 1 to 2 weeks. He went on a vacation, and he came back from the vacation. At that time he saw that on his plate, which was having the staphylococci, instead of cephalococci, he found out that there were going to be many other type of organisms which were grown over them. So which other type of organism was going to be present? So he wanted to know that his plate was contaminated by which type of organism, because when he carefully observed those plates, he found out that the new organism that was growing, which was a dark colony which was growing, it inhibited or it stopped the growth of the staphylococci. And when he carefully observed it, he found out that it was a fungi, it was a mold, that was Penicillium notatum. So this Penicillium notatum, a fungi, had contaminated his plate, had overgrown on his plate. And when he carefully examined, he found out that this Penicillium notatum stopped the growth of staphylococci. So he then started knowing what is going to be the substance in the fungi which is not allowing the growth. So he extracted the chemical, that is the penicillin, which is produced by the fungi, that is Penicillium notatum, which is helping to kill the other bacteria. And hence the word antibiotics came. It was killing the other bacteria. A fungi product, penicillin, was killing the staphylococci. So he extracted the penicillin, and hence the discovery of the antibiotics was possible. Later, more discoveries took place, and Ernest Chain and Howard Florey established the full potential and effectiveness of the antibiotics. And finally, in 1945, this Alexander Fleming, Ernest Chain, and this Howard Florey, all these three were awarded the Nobel Prize for the discovery of antibiotics. Then, let us see some of the chemicals, enzymes, and other bioactive molecules which are produced on an industrial scale, on a large scale. So, we can see first, let us consider the organic acids that are produced from the microbes. So, here are some examples of acid producer microbes. So, some examples are Aspergillus niger. It is a fungi. Aspergillus niger is a fungi, and after fermentation it is producing citric acid, which is commercially important. Then Acetobacter bacteria, it is producing the organic acid, that is acetic acid. Clostridium vitacillum, it is also a bacteria, it is producing the butyric acid. And lactobacilli, again a bacteria, is producing lactic acid. So these are the names of some or microorganisms which are producing the organic acids. So this is going to be important. So this can be asked in the exam. Name the microbes which are going to be producing the organic acids, or name some microbial products name some industrial products which are going to be the chemicals, enzymes, and other bioactive molecules. So the Aspergillus niger producing the citric acid, Acetobacter aceti producing acetic acid, Clostridium butacillum producing butyric acid, and lactobacilli producing lactic acid. Sometimes even it is asked for 1 mark, name the microorganism which produces citric acid. Your answer has to be Aspergillus niger. Name the organism which produces lactic acid. So it is lactobacilli. So these type of questions can be asked on this. Then coming to the alcohol. So let us see which alcohol, in the production of the alcohol we have seen even before, the yeast, that is the Saccharomyces cerevisiae, is used to produce the ethanol. Then we can see the enzymes are produced using the microorganisms. Examples of enzymes can be the lipase. Lipase is going to be used in detergents, in the making of the washing powders, that is your detergents. So used in the detergent formulations, and this lipase in the detergent is helping to remove oily stains from the laundry. Laundry is nothing but from your clothes. From the clothes, if the oily stain has to be removed, then the lipase is going to be helping. Then we can see that the pectinase and the proteases enzymes are used to clarify the bottled juices. So whatever the packaged juices you are getting, you can see they are clear, they are transparent. So these pectinase and proteases enzyme produced from the microorganisms are used to clarify the bottled juices. Then, another important enzyme, the streptokinase. The streptokinase is also known as the clot buster. So, this streptokinase is going to be produced by using the microorganism that is a streptococcus, again a bacteria. So, what this streptokinase is doing, it is going to be helping to remove the clots, the blood clots that are going to be there in the patients who are having the myocardial infarction. Myocardial infarction is nothing but minor heart attacks what we get, or the blockage is going to be there. As you can see here, this is going to be the arteries from where the blood is going to be supplied to the body parts or carried from the body parts to the heart. It is through the blood vessels. So if there is going to be blockage here into the blood vessels, which can lead to myocardial infarction, that is some problem in the heart, beating of the heart, then these clots that are going to be there, they can be removed by using the enzyme that is the streptokinase, and hence the streptokinase is going to be also called as the clot buster. So this is the streptokinase, again an important enzyme. Then other such products are going to be medical products, you can say the cyclosporin A. So cyclosporin A that is there, it is going to be produced by the fungus that is Trichoderma polyspermum. The fungi name is Trichoderma polyspermum. It is producing the cyclosporin A. For what this cyclosporin A is used? It is used as an immunosuppressive agent in organ transplant patients. Now what is happening, in some patients you need to replace certain organs. For example, in some patients maybe the kidney failure is taking the place. Both the kidneys are not working. At that time what the doctor suggests? Kidney transplant. That is, you have to put some other person's kidney, one of the kidney, into the patient body. So at that time, the patient that is there, it will not accept the kidney easily, because the kidney does not belong to his body. So what it will do? Body will start producing the immune cells. You have studied the immunology. So immune cells, it will start rejecting, or it will start killing the foreign body that is going to be there into our body. So it is the nature of our body, which is the work of the immune system of the body. So what this cyclosporin A is doing, whenever there is going to be such type of transplant, until the body recognizes that organ, until then it will suppress the immune activity. So it is acting as a immune suppressive agent. It will stop the action of immune system for some time till the patient gets acclimatized with the kidney that is transplanted to him. So that is the cyclosporin A. Then next is going to be the stannins. So stannins are going to be produced by a yeast. It is a type of yeast, again a fungi. So Monococcus purpurus. So Monococcus purpurus, it is used as blood cholesterol lowering agent. If you are having high on cholesterol, then this type of the product of these type of organisms can be used to lower the blood cholesterol. How? It is doing this by inhibiting the enzyme responsible for the synthesis of cholesterol. So in our body there are going to be the enzyme which is going to be producing the cholesterol. So this product, that is the statin, can be used to lower the blood cholesterol by inhibiting or stopping the enzyme activity which is responsible for the synthesis of the cholesterol. So when the synthesis is going to be stopped, automatically the blood cholesterol level also going to be lowered. So that is going to be the statin, a product of the microbial activity, that is microbial organism, that is your Monococcus purpurus. So this is going to be all about the microbes that are going to be used in the industrial products. So in the industries, they are going to be using the microorganisms to produce different type of products which are helpful to the human beings. Maybe it is the beverages like your wine or whiskey, or whether it is going to be the antibiotics like penicillin, or you can say the enzymes and clot busters and the statins. All these are going to be produced with the help of different type of microorganisms. So this is going to be all about microbes in the industrial products. In my next video, we will see how these microbes are playing an important role in the sewage treatment. So microbes in sewage treatment will be my next video.